Bismillahirrahmanirrahim. Myself is Professor Dr. Saima Masood, working as a chairperson in the Department of Anatomy and Histology at University of Veterinary and Animal Sciences, Lahore. Uh, the study of my topic of my study is histomorphological effects of deep dry needling on myofascial trigger points in skeletal muscles of Mr. rats. So what is actually myofascial trigger points is described as a pain caused by myofascial pain is pain caused by myofascial trigger points in skeletal muscle fibers. It is common in regional musculoskeletal pain syndrome, either alone or in conjunction with other pain categories. So this study was aimed to address the lack of comprehensive data on the histomorphological effects of deep dry needling on myofascial trigger points in visitor rats. So our study could potentially improve treatment protocols for myofascial point pain syndromes and advance understanding of cellular mechanisms managements. It also focused on histomorphological changes of deep dry needling in skeletal muscle fibers and myofascial trigger points. So what is a trigger point? Actually, the trigger point is made up of multiple contraction knots. An individual contraction knot is muscular fiber and with the fiber segment that has severely contracted sarcomeres and larger diameter. So, severe localized sarcomere contraction zones have been observed in both animals as well as in human beings. Our study was actually in our study 40 male and female visitor rats were taken and divided into 4 groups, having age between 6 to 7 weeks. First group was control group and a negative control group. Second was deep dry needling group, and the third was trigger point group. And induction by the neostigmine at the rate of 0.1 per kg body weight. And group 4 was actually having both myofascial trigger points and deep dry needling induction. 30 minutes post neostigmine methyl sulfate administration, cholinergic syndrome was uh, clearly seen with the sign and symptoms. So each needling session consisted of giving 15 approximately 10 to 15 punctures in and out positioning movements to release the trigger points by local switching response. So our results regarding histomorphological analysis of gastrocnemius muscles indicated um, how we process the slides that by taking samples of gastrocnemius muscles and then fixation in buffered formalin solution and grading ascending grade using ascending grades of ethanol for dehydration purpose, and later on we did microtomy, and after staining with standard hematoxylin and eosin protocol, we analyzed the muscle fascicles under microscope. The muscle here you can see the micrograph indicating different groups, muscle groups in different studies. First is the control group, and A is indicating muscle fiber, while B is muscle fascicle, and C is endomysium, which is a connective tissue layer around the muscle fiber, and perimysium along with the connective tissue covering along with the muscle fascicle. So you can see the description of results here that G1 group is actually having normal saline, and G2 group deep dry needling group, and the third is G3 group, which is trigger point group, and fourth group is having both trigger points and deep dry needling. Then histomorphological analysis of gastrocnemius muscle was done, and not only using H and E staining. You can see the previous slides were at 10x, but these slides are at 40x objective lens. And these are the different histomorphological analysis. Later on, we used Mason's trichrome staining method to indicate the connective tissue or the collagen fibers around the muscle fascicles and around the individual muscle fibers. We can see that in third group, this is localized. This greenish color is indicating the collagen fibers in between the muscles, and in the fourth group you can see that this localized collagen fibers, they were moved around they were dispersed at the periphery, so which is indication of the um, recovery phase, just close to the normal. So these are the different scales and by using these scales, which are used in rat Grimace score scale, we can see that how the pain can be relieved. And these are the different facial expressions of the rats during these induction of the trigger points. At in first in first picture you can see that this is normal, and the uh, second picture is telling you the facial points, facial expressions of the mouse with the dilated eyes, and its ears are at the periphery. And our study is indicative of the that deep dry needling, not only the myofascial trigger points, but this both combination of deep dry needling and myofascial trigger points are significantly controlling. They are significantly contributing to the normalization of the morphometric parameters, and they are helpful in decreasing the they are helpful to increase that pain, myofascial pain, and these all findings are very much obvious with the histological findings. And we can see that deep dry needling improves the histomorphological parameters and reduce the pain, myofascial trigger points, supporting its potential as well as it is effective for therapeutic approach for the myofascial pain syndrome. Thank you very much.